and I'll be presenting on the CFDE collaboration between HuBMAP and Kids First. Our goal in this project was to develop a knowledge graph to integrate common fund data sets at the level of the data points. This would allow biomedical researchers to ask questions of this interconnected data by utilizing simple, complex, and artificial intelligence queries and analyses. This project was only made possible with common fund data, ecosystem activities, and support. This knowledge graph currently contains over 50 million data points and about 150 million relationships between those data points. This allows researchers to ask complex questions that they may otherwise take months to put together and realize. For example, a researcher may suspect that some heart defects could be due to dysregulation of glycosylation, leading to timing changes in developmental programs. They would like to know which glycosylation sites and which glycan types could be disrupted on specific proteins related to heart defects, so that they can then go and query Kids First genotypes for these possible sites. To make this kind of query off of Kids First data sets and off of Glygen data sets, we can take a Kids First phenotype here, represented by HB001631, which I believe is an atrial septal defect, and we can simply query the engine with this query asking to find us all proteins that may have this phenotype. And here's this query, all the proteins that support this phenotype. So here's a gene associated with the phenotype of atrial septal defects. Here's a gene, one particular gene. And then here's a particular glycosylation site, one of the glycosylation sites on this protein. A further query for this could be done to return all glycosylation sites on all proteins that's shown under here, which I don't have time to show today. But if you did this query, you would get all glycosylation sites and all proteins associated with this particular phenotype. A second question would be, if a researcher wants to check all cis-EQTL sites for possible germline variants changing gene expression in Kids First patients that may contribute to a birth defect or a cancer phenotype. Given a particular subject ID in Kids First, we would like to find all the HPO terms for that subject, and then find all genes associated with those HPO terms, and then find all cis-EQTLs and GTECs that may regulate those genes' expression. We have to note that this query is not going to return variants from Kids First, but rather potential genomic locations to test for variants. So here's a patient ID in Kids First. We're going to ask the engine to find us all the phenotypes from that child, all of the genes associated with that phenotype that have been annotated in the public domain. Then find us all the cis-EQTLs associated with those genes, and then do so and return all the locations of those EQTLs, so that could be then used for a query against Kids First data. And I'm going to limit it to 100 because there's going to be a lot of results. You can see how quickly this query executed. So researchers now have the ability to query Glygen data, GTEx data, and ostensibly Kids First data to get data back to inform research on specific sites that may be functionally related to heart defects through gene to phenotype relationships. Another query would be starting with a compound. Now we're going to move into compound relationships against, compounds like chemicals that can affect gene relationships, gene expression relationships. So starting with a compound that affects gene expression from links, find all the affected genes in GTEx with those genes with an expression value greater than some sort of user-specified threshold, and then return the tissues where those genes express from GTEx. So if we do this query, here we're going to use this compound called mosapride, and we're going to find all of the tissues where those genes express and what tissues may be affected by this compound. Because if this compound affects certain genes in certain tissues, we can expect to find the tissues that are affected by those genes, that are affected by the compound. So it takes a second to do the query, but here you see how quickly we got the results back, and it's only showing the first 1,000 rows. But here's all the tissues that could be affected by this particular compound, because they have a significant amount of expression of the genes that this particular compound affects. So here's the expression level, and here's additional information on what tissue this gene expression was found in. So extended research queries could be done, linking that back to, for example, Kids First phenotypes, by linking those genes back to phenotypes, for example, if you were interested in reproductive toxicity. You can also do additional analyses using HuBMAP and try to find particular cell types that express that gene and are expressed that could be affected by MOSA pride. So that is essentially our quick and dirty presentation for you. There is some background information here and on all the different types of data that's in this knowledge graph and more supplemental info. If you want to contact me, my email address is taylordm@chop.edu, or you can also access this page here at this location and contact us through GitHub. Thank you very much.